Good afternoon. This is Dr. Dan Guerra coming to you from Authentic Biochemistry Studios in the inland Pacific Northwest. Today, what we're going to do is begin my in-depth discussion of accumulating all the previous arc of aging lectures and immunology lectures and doing a recombination so that we end up having a dialectical event ontology of the association of the immune system with aging. As my resource, of course, I'm using the primary scientific peer-reviewed literature out of journals, and most of these journal publications are very recent, but when it is necessary, I go back into the stacks and we'll pull papers from the early 2000s, and then anywhere from then all the way up to the present, even papers in press. That's what I do if you've been watching and listening to Authentic Biochemistry and Verimed lectures for the last couple of years. So I'm not going to belabor that point, and we're just going to go ahead and get started with it. So let's do a slideshow. Okay, let's move me out of the way here a little bit. So I'm going to call this the Immuno Diaeventontological framing of the aging human, Lecture 1. Today, as I said, is the 1st of October, and I presume this will take me at least 2 lectures, possibly 3, depending on how long each one of them is. Okay, so I'm not going to really spend much more time developing authentic biochemistry and explain to you what I do here. If you've been watching me over these years, you know what we're up to, and obviously I would like you to subscribe to my YouTube channel and also to my podcast, Authentic Biochemistry. If you haven't done so, please do so at your earliest convenience. And also, please support us if you can by donating either to my Patreon page or directly to the podcast site. And I appreciate your attention and your interest in authentic biochemistry. Okay, so what I want to do today is go back to the foundation of what I'm trying to do when I give you these lectures. Particularly when I choose a large subject area like aging, cellular senescence, and I explained to you at the beginning that I'm doing this because I want to draw attention to how the immune system or some other great system in biochemistry has a direct and indirect association with a physiological process. And of course, you also know that I do a lot of pathophysiology, and maybe many of you think that immunology is all about pathophysiology, and my argument is that it is not. That even if you bracket off the concept of surveillance as an important component mechanism of immunology or the immune system response, that's not even what I mean. I'm talking about the immune system actually having a role in development and differentiation, including the central nervous system, including the brain, and then the product of the brain, at least at the phenomenological level, the mind. Okay, so let's start off with the dialectic. And what I will say is that the genome contributes to heritable phenotype of the central nervous system. Let's get rid of that if we can. Some nasty ads. The environment contributes to the heritable and non-heritable phenotype of the CNS. So you got the genome, you got the environment, then you got what I'm calling the epigenomic transcendency. And that exerts control over gene expression, obviously, and it acquires and extends neuroplasticity and elasticity that result in disease, normal health, and eventually the processes that control cellular mortality and lifespan. So the immune system very basically is broken down into lymphoid cells and myeloid cells, as shown here in this slide. Myeloid cells first, you have the granulocytes and the monocytes. The monocytes give birth to macrophages, Kupffer cells, dendritic cells, and there are more, but that's a basic lineage. And the granulocytes give birth to neutrophils, basophils, and eosinophils. These are your basic innate immune response cellular armamentarium. Lymphoid cells include, of course, the T lymphocytes, the B lymphocytes, and the natural killer cells. And we've talked a lot in the last month and a half on T cells. I don't spend much time on B cells. I've done that about a year and a half ago. I gave a series of lectures on B cell physiology, pathophysiology, biochemistry. And we can go back and do that. But right now, I don't think I need to develop this Diaeventontological perspective on aging, because T cells are what basically control most of what the B cells are up to. That, and of course, in coordination with antigen presentation and with complement. So T cells give rise to the so-called helper cells or effector cells, suppressor cells, also known as Tregs, and the cytotoxic T lymphocytes, which we've been spending some time on very recently on the Authentic Biochemistry podcast, right? All right, so I think that covers your basic immune system. Now, why am I doing chemistry? You'll see in a moment. The valence shell electron pair repulsion concept. I'm giving you a case in point where you have a trigonal plane. Now, there is CO3, right? And there's a formal -2 charge, right? So let's take a look at this diagram and follow along why I'm showing you this, so I can get you into the way that I like to examine biological systems. So VSEPR allows that electron pairs will repel each other, whether they are bonded or in lone pairs, because of the electronegativity of the electrons. Okay. VSPR then will result in a molecular geometrical arrangement that minimizes repulsion. Molecules that contain a central atom, for example, carbon, bonded to 3 oxygens, shown here, should then be restricted to a 2-dimensional or trigonal, which is just another way of saying triangular plane. Such a molecule shown here is carbonate, of course, where 1 oxygen is double bonded to the central carbon, shown here, and the other oxygen atoms are single bonded. Okay. Now, why am I talking about chemical bonding when I just introduced to you a dialectic in biology? Well, remember, I'm a biochemist. So I don't just talk about biological systems like physiology and genetics. I also talk about the chemical aspects of living systems. That's why I am called a biochemist. That's what I do. So we take 2 major disciplines in the natural sciences, chemistry and biology. And of course, chemistry and biology themselves are built on things like physics, and physics is built on mathematics, and mathematics is built on logic. So this goes way back all the way to the precursor of scientific endeavor, and that is philosophical meanderings. So biochemistry, as I see it, is one of the top drawer sciences for anyone who wants to study biological systems, because it allows us to understand at multiple levels of integration how living systems operate in real time. Because biochemistry is about event ontologies, not substances. And you might say, now, wait a minute. Aren't all those biochemicals like acetyl-CoA and malonyl-CoA and serine and, I don't know, let's say cholesterol, aren't those all substances? Well, yeah, they are. They are chemicals. But when you start talking about biochemistry, you're not talking about test tubes anymore, except, of course, we use test tubes or actually microfuge tubes all the time, and sometimes borosilicate glass if you're a lipid biochemist like me. But we isolate from living systems as a biochemist, and we put them into tubes. We isolate, fractionate substances from living systems, and we study them. But ever since really the genetic code was determined. And we had a clear understanding of how to do molecular genetics, first in bacteria, like E. coli and lactobacillus, and then moving through model organisms like Drosophila, which had already been brought in the 1920s with the molecular genetics program without the nucleic acids, if you will. And of course, yeast is a model system. And then we went to mammals, in the late 50s, early 60s, using mice and rats, and still use mice and rats. But in the last 20 years, almost 30 years now, many biological studies can be performed with humans. Now, these are often pharmacotherapeutic type of studies where there's a disease involved, that people have put on a series of different kinds of medications with different modes of action, right? And those different modes of action then can be studied only in the living system. And of course, what you're looking at is the output of that pharmacotherapy is some recovery from some disease, right? Now, pharmacotherapy is only one way to counteract disease. One is, of course, surgery as well, very important. And also other kinds of therapy, like, for example, physical therapy, and changes in diet. Now, all of those, surgical procedures, diet, for example, those actually do involve changes in biochemistry within the system. That is, if you're looking at the human body, it's the human body. So everything fundamentally rests on the biochemistry of the system. You could say, well, isn't it just the chemistry? And I would argue no, because the chemistry embedded in biology is not just chemistry. It's a completely different thing. It's like saying that when you look at a cow and you say, well, that's a steak. No, a cow is a living organism that is able to break down cellulose and hemicellulose because it has multiple stomachs. It's polygastric. And so that it can break down those beta-1,4 linkages, whereas other mammals can't, and they can turn grass and alfalfa into digestible matter that ends up becoming protein. And the protein becomes the muscle, and the muscle is what becomes the steak, right? So you can't say that because a steak, a finished product, comes from a cow or comes from cattle, that they're the same thing. One is derived from the other. Well, the analogy isn't necessarily the best because certainly biochemistry isn't simply some portion of some larger thing. It actually is everywhere. It's omnipresent in every living system, right? And so, being a biochemist, I've been profoundly lucky to have landed in this discipline of science, because once I got into it, I realized that this is the most challenging, at least from my perspective, and the most interesting, also perhaps just at least from my perspective. Because in order to understand biochemistry, you have to have a really good understanding of chemistry as well as physics, at least at the level of thermodynamics and how things change through time, and how energy and matter are and interconverted, excuse me. But also you need to understand that all embedded in a biological system. So that means it's dynamic, real-time event, always with biochemistry. Okay, so back to the slide. Why am I talking about chemical bonding? Well, chemical bonds are not merely material substances, I argue. Chemical bonds are representations of dynamic energy confluence. Dynamic energy is obtained temporally and is therefore an event rather than simply a substance. Event ontology therefore takes into account the dynamics of, for example, the most sophisticated of the human system, neural circuitry. Neural circuitry obtains via immuno-epigenetic phenomena. That means neural circuitry is the result of. When I say it obtains, that means this is where it comes from. Right? And I've shown through various research that I've done on my own in laboratory and published on, as well as in several review articles, that it's been well documented, not just from my laboratory or from my writings and my perusal of the scientific literature, but also from hundreds of other laboratories worldwide, that indeed neural circuitry does obtain in no small measure from immunoepigenetic phenomena. And if you don't know what epigenetics is, don't worry, we will go back and discuss it. But basically, it involves mechanisms whereby certain genes are expressed in certain tissues at certain levels, at certain times, which differentiates one cell type or one tissue type from another. Right? And the mechanisms for epigenetics include things like methylation of DNA, acetylation, and even methylation of histones bound up in condensed chromatin, and of course microRNAs, which can be used to control or silence at the messenger RNA level the final translation, therefore what we call expression of a gene, ultimately to a polypeptide. But it's much more than that. Epigenetics also involves all the enzymes that tailor all of the membrane lipids. And membrane lipids play a very vital role in the development, differentiation, and homeostasis, and the continued event ontology of the living system throughout its life. And if it's a long-lived organism, like a human being, right, 70 at the minimum these days, that means it's a very long process. And the process doesn't stop at puberty. It doesn't stop in your 30s or middle age, or even when you get much older. The process continues until the system actually stops existing when it dies. And that's because your brain is constantly involved in learning until it no longer functions. Right? So that's something to keep in mind. So the trigonal planar model, which I just showed you for carbonate, is one I chose because I think it's the simplest way to grasp, apprehend what I'm trying to get into with my Diaeventontological perspective. So the trigonal planar model is the simplest representation, what I would say, the simplest. It's not necessarily the most accurate, but it's the simplest one to get a handle on for neural system dynamics. It's my argument anyways. So here's what I've developed now about 3 years ago. I call it the Diaeventone. And you can see the three components of it. You can see that it is trigonal planar, and it is planar. You have the genome here, you have the environment here, and you have the immunoepigenome here. And the coloring is important because the immunoepigenome is going to have components of the genome and components of the environment because the immune system responds to the environment, right? In terms of, like, say, stress. For example, like an etiologic agent like a parasite or a virus or a bacterium or simply an inflammation, right? So let's go through what I wrote here when I first put this together. Neural networks are processes becoming eventual in nature as opposed to substantial or only substantial. In fact, it's not even a good description of it. So an event ontology that reaches across the neural network of genetic and environmental interaction, that's what I'm showing you here, is articulated and synthesized through epigenetic modifications of the classical immune system, thus creating a responsive plastic, which means the change is made and it stays, and elastic, that means a change is made and then it is repealed, memory field capable of learning, remember I'm talking about the central nervous system, ideation, imagination, and understanding through time. The pattern of events appear as energetic fields, because we're talking again about biochemistry. So you're going to have a conversion of certain nutrients, let's say proteins, or let's say carbohydrates or lipids, and because of the fact that you can interconvert matter into energy and energy into matter, of course, we all know that principle. We can derive energy because of the reducing equivalents generated from the oxidation of some of those foodstuffs, particularly carbohydrate and lipids in the form of fatty acids, but also to no small extent, amino acids derived from proteolysis from proteins that are ingested in the diet. Okay. So through time is really important too. That's why it's an event, right, and not a substance ontology, as I've been trying to argue. So the pattern of events appear as energetic fields rather than particles of matter, right? Obviously, much like chemical bonds. Chemical bonds, again, are not substances. They're energetic interactions between atoms or among atoms within a molecule, right? And usually these molecules can be somewhat extensive and large in terms of their complexity when you're talking about biochemistry. So another thing I want you to keep in mind here as we get into this paradigm, central, individual, agentically interacting, this diaventone, through the events obtained across the inherited genome, the changing environment, always changing, right? And the transcendental, that doesn't mean anything spooky and otherworldly. Transcendental just means the means by which the whole system comes together, right? The transcendental immunoepigenome through time. Okay, so that's the paradigm that I generated I guess 3 years now. And I've published on this, and I've given lectures at various locations on it. So hopefully you've caught some of that. So here's a combined effort, Diaventontology, an adaptational, processive accounting of the rational human mind. And understand, see where this is, these are the original slides I used when I first presented this at an international meeting. Now, why am I talking about the human mind? Because I was working in neuroscience at the time. And that's what I was trying to put together. How does the brain generate mind? Right? And again, this is all phenomenological. This isn't dealing with anything beyond what we can discuss in terms of our senses, right? There is, I think, a strong noumenal component here, but that's not what I'm doing here because we're doing science, not philosophy. You want to listen to my philosophy? Go over and listen to some of that. But this is now my science side, right? This is all phenomena, right? And so the reason I want to talk about the human mind is because it's the most complex. If we can understand a better way to describe the human mind from the brain, then I believe we will be able to understand pathophysiology starting in the central nervous system, something that's really important to humans. Think like neurodegeneration, Alzheimer's disease, Parkinson's disease, things like that. But then also all the other pathophysiology in the body, say the cardiovascular system, or all the cancers that can generate peripherally and centrally in the human system. Right? And then maybe the most important part of this is how does the human learn? What is the event ontology of learning? How is knowledge actually gathered and stored? And not so much that, because to me that's trivial. What really matters is how can you integrate a network of knowledge to be able to understand something as complex as biochemical systems, right? And then be able to call upon that using your mind rather than just looking it up online all the time, right? Your mind is the process, it's the synthesis of all the information. And it doesn't become knowledge until after that a priori categorical induction occurs, and you're able to synthesize the manifold of all the incoming, which involves sense data. So there is a rational side to this, and there's also an empirical side to this, if you want to consider it from the epitope of philosophy. So the immunoepigenomic induction of neural endocrine. So now I'm listing the different systems because I'm a biochemist. So I'm going to give you some florid account of it. The induction of neural, endocrine, and metabolic response to the micro and macro environment, micro meaning internal, at the small level, the molecular level, even the atomic and subatomic level, right? Quantum level if you want. And the macro environment, that is what's outside of you, right? Everything, because you're a heterotrophic organism, so it's also what you eat and what you breathe and what you drink. Through classical constitutive surveillance, that's part of the immune system, particularly the innate immune system, but also, of course, T memory cells and B cells, plasma cells in particular, and the acquired effector or helper cellular and humoral defense stratagems, which are lymphocytes, using reversible covalent modification. This has to do with the epigenetic reprogramming. And hydrophobic interactions, this has to do with membrane interactions, very important for thermodynamics, bioenergetics, as well as transducing signaling phenomena of nucleic acids, proteins, and lipids. Okay. Carbohydrates are also important there. I didn't add them there because I'm trying to make you think about the major players in the central nervous system when it comes to signal transduction. Of course, carbohydrates are absolutely important because they're often used, glucose in particular, as the bioenergetic fuel. So I'm hoping that diaventontology someday, long after I'm gone, very likely, will become the central, or could be used as a central molecular theory for free will driven, because this is the really important issue here. As a living system, as a human being, what we do is what we choose to do because we have free choice of the will. Now, if you don't buy that part of my epistemology, then you don't have to. Diaventontology still works. But what I find so intriguing is that all events that are associated with what occurs around human beings does seem to be events which are caused by either directly or indirectly, that is through pure reason or pure empirical phenomena, the agency of the human mind. The agency of the human mind, the endeavor through time until that human mind is laid to rest. Okay? And so that's why I'm very interested in this free will driven, agency based, existing individual adaptation, that each individual is unique. And as a biochemist, I can tell you that that is absolutely true. No human being is identical to another human being. Identical twins are not identical. They have different patterns of gene expression. If you want to think of it at the simplest level, it's because each cell is not at the exact same stage or age of development in any system, in any tissue bed, even in identical twins, even with the same genome. But it's the epigenomic fractionating of all of that system over time, which generates, again, an event ontology that results in one individual being unique from every other individual. And even the same individual, biochemically and physiologically, is unique from that same individual at some earlier time. Okay. That's how a biochemist thinks, right? We think about events. We don't just think about what's this here now, right? We think about how processes are understood as foundational and coherent with living systems. That's why it's so important to have some handle on thermodynamics. So, individual adaptation and knowledge acquiring epistemology that explains, or at least helps to explain, the core event ontology of human existence. So that's why I wanted to develop this. I know it's a really huge brush I'm using in this portrait framing here, but I'm doing it because I figured that if I can bring it as a large theory, then I can use that large theory to generate and populate it with any number of numerous hypotheses, which via deduction I can use to generate experiments to look at systems with the consideration that they are undergoing events in real time and in a process of time which has an earlier phase or present perfect phase, a phase which is immediate, and then a phase which is mediated. Right? And so that allows me to cover all that ground. Okay. So here was where I actually went through an entire process to give you a diaventome of a particular disorder. So this comes from the end of about 10 lectures I gave, probably more than that, but at least 10 hours of lectures on autoimmune diseases, which I started way back in December of 2019. And I carried through all the way through January and February of 2020, and even into March before I started doing other lecture series. So covering a lot of ground, looking at peer-reviewed papers, I developed what I call the autoimmune neuroinflammation Diaeventontology. Let's take a look at it. You have the genome, and the genome we determine, now this is again the result of various hundreds of papers that I looked at, various genes involved, MDL, BCLX, neutral sphingomyelinase, ceramide synthase, all the folic acid metabolism. Now, these are specifically what I found in the literature associated with autoimmune diseases, which have a linkage to neuroinflammation. That's why they're very specific, you see, because these come from the scientific literature. I didn't just pull these out of a biochemistry text. I discovered that these are the things that came up when I looked at that literature on autoimmunity, neuroinflammation. Right? That's what this comes from. Study, long-term scholarly work to get to this. Methionine metabolism, which of course is associated with folate. Serine metabolism, likewise. Fatty acid metabolism. Glycerophospholipid metabolism. And sphingolipids. So basically covering a lot of ground of biochemical pathways. The environment, what we determined with all those lectures. And you can go back and watch them all and listen to them. They're all on my YouTube channel or in my Authentic Biochemistry podcast series. The environment that we keyed in on all these papers were including, remember I told you that environment, you have to get a larger mind about what I mean by environment. So a temporal existence or aging or senescing through time is part of your environment, even though it's a process, because everything here is an event, see? Aging, inflammation, infection, which are separable, of course, but which can co-occur. Stress, this can include intellectual stress, emotional stress. Sedentary lifestyle, we found a lot of papers talk about sedentary lifestyle for autoimmune diseases. Linked to that, probably, often, obesity, alcohol and drugs. Drugs can not just be the recreational drugs, but also pharmaceuticals. We talked a lot about that in this complete discussion of the autoimmune response from the literature. Neuropsychiatric disease, let's say major depressive disorder, let's say bipolar disorder, even schizophrenia, we covered that ground. And then, of course, membrane lipid turnover, because that is part of the environment. The cellular environment is all membranous when it comes to a discussion of signal transduction cascades, right? and communication. So what was the immunoepigenome component of this? Microglial activation. Now, we're going to talk about that in this series of lectures here in full florid attention very soon. But microglia are kind of the resident macrophages or innate immune cells in the central nervous system, but they're much more than that. B in plasma cell recruitment. Okay. T cell differentiation and modulation. So T cells started from CD4 positive, CD8 positive, naive T lymphocytes, all the way to generating Th1, Th2, Th17, Tregs, for example, natural killer cells. Right? All of this was discussed. The production of a whole family or constellation of cytokines and chemokines, growth factors, T-resident memory cell activation, which was another important issue in autoimmune disease, and the proliferation of those T memory cells upon induction, and then T cell programmed cell death. And that's like the PDL1 or CTLA4 that you hear about with checkpoint inhibitors in cancer research. Right? We found that nesting very comfortably in the higher branches of the autoimmune neuroinflammatory peer-reviewed scientific research literature. Other things that were involved were microRNAs, as I said, that's part of epigenetic systems. Sirtuins, which have to do with the acetylation pattern. So you have sirtuins and you have HATs. HATs are histone acetyltransferases, so they add acetate to chromatin. When you have acetate in chromatin, you make heterochromatin, and that makes it more likely to have expression. When you have a sirtuin, that's a deacetylase. And so, euchromatin versus heterochromatin. When you deacetylate them, you collapse the chromatin. When you collapse the chromatin, you holistically, generically make the chromatin less likely to be expressed at the level of transcription, making messenger RNA or even ribosomal and transfer RNA, but also for recombination and repair. Right? Condensed DNA is much more inert. Sirtuins, which are deacetylases, which are enzymes that you find throughout the body, and we talked a lot about sirtuins in the past, can then regulate globally gene expression. Of course, these are epigenetic phenomena, but they also cohere with the immune system, and they are, at least at some level, directly related to the genome, because the genome is where you find the genes for sirtuins, for example, and the environment, which can control their induction and turnover, say proteolytic degradation via the proteasome. And then there were the DNA methyltransferases and the DNA methylases, the ones that add methyl groups to cytosine residues on CpG islands, proximal as promoters to genes, and chromatin is going to be remodeled for gene expression. And then again, so you have methyltransferases, you also have those proteins, those enzymes, which remove the methyl groups. Okay. So that's what we found using my Diaeventontology paradigm while looking at neuroimmune, neuroinflammatory diseases. So living systems, right? You can discuss them using the square of opposition. So I know I have a triangle there, and I talked about trigonal planar, but it doesn't matter. There's geometries everywhere. I mean, it's natural, right? Ask Plato, ask Pythagoras, for that matter. So, and I have to wear my glasses because of the font on the side of the slide. Living systems interact according to a pseudo, that's what this is, three-dimensional biological trigonal plane, which is three-dimensional plane, obviously, of opposition. And I'm thinking about the square of opposition. Now, why am I bringing this up? In order for you to take a look at the scientific data, the evidence, take it from data and articulate it into evidence, which can be used to generate what? Discussion of concepts, an increase in understanding to come up with finally a new hypothesis via deduction. Right? The scientific method that I just described to you in the reverse, actually in both directions, which is the best kind of science. So let's take a look at the squares of opposition, the four corners. You have universal affirmatives, no harm to an individual, maximum benefit, rare or occasional epistasis, that means interaction of genes. Universal negation, severe harm to an individual, 100% epistasis, a lot of gene interaction. It would have to be, right? Because then you're taking the one system and asking, in any possible permutation, how is it affected? Right? And if it's universal, it has to be affected negatively. Particular affirmatives, some benefit to the individual. Disinterested epistasis, that means it's like the monads that were described by Leibniz to say that these are windowless modules of living systems that interact, but they interact in a way that they have their own agency, yet the interaction itself generates a new agency. Right? And then ultimately, particular negation. Some harm to the individual, predicated epistasis. That means that the harm is caused or it's predicated by some kind of gene-gene interaction. Gene product is what we're talking about, but you can think about lipid membranes too. So why did I give you this square of opposition? Because you can take the information when you read it in the scientific literature, and you can come up with arguments that will generate things like, give me all SRP. So to deliver that into a specific all aging is detrimental. That's a universal affirmative, even though that sounds like a negative. But you could say for the universal negation, no aging is detrimental, because maybe you realize that aging allows the full lifespan to occur. Right? So, but this involves, you see, biochemistry and pathophysiology and normal physiology, because you're still talking about a biological system from a biochemical point of view. Right? So you can still talk about negations and affirmations. And then much more commonly, you have particular affirmations and particular negations, right? In that system, some aging is detrimental because it may allow you to pick up infections more easily because your immune system is more quiescent because you have more T memory cells. That's one particular derivative of my study of T lymphocytes that I found out, and you hear occasionally. And then a particular negation, right? Which could mean that some aging is not detrimental, right? And again, an easy one to put in there is all of the biochemistry and all the molecular physiology associated with generating a new memory, like this lecture that you're listening to. All right, so that allows me to get to, so now that was a big introduction, or the word I like to use, of course, is prolegomena. And I just like that word. It's a word that I picked up reading Immanuel Kant. Right? So all of that is just to lay out where we're at right now, where we left off in the Authentic Biochemistry lectures. So let's take a look at this paper here. It comes from the Journal of Neurochemistry, 151, issue 6, published in December of 2019. Those are your page numbers. That was published. That was the first time it was published. It didn't come out really until 2020. So let's take a look at what this paper is going to tell us. I broke it down. I know there's a lot of writing here, but again, it's so that when you go back and listen to these lectures, you don't just have to listen to me. You can stop and you can read it and you can take notes. Because remember, I started out doing all of this as over 30 years of teaching in a classroom. So I like my students and my colleagues and anyone else that's interested to be able to get as much information as I can obtain for them and distribute it. And this is the way I do it on this format. So human aging results in a gradual loss of normal functioning. Well, I think we're pretty much on board with that. Changes at the cellular level are believed to contribute to that organismal level, to that whole process, right? So those are, I think, good hypotheses. And there's a lot of research that suggests that. You don't have to own them. You have to say that that's a universal, but you can say it's a partial. Aging within the human brain is of considerable interest because age-related changes are frequently irreversible and can lead to changes that severely impair normal activity. Such a thing as selfhood, the most important, for example, I think. Selfhood, if you start losing that, like in Alzheimer's disease, that is the most horrific and scary thing for a loved one. Furthermore, or you can think about motor skills, right, as you age, or sarcopenia as you age, right? Loss of good muscle tone, sarcopenia, muscle senescence. Furthermore, the changing cellular environment of the aging brain is the single largest contributing factor to increased likelihood of developing these neurodegenerative diseases that I just mentioned to you, like Alzheimer's. Okay? So this is all coming from this paper. They're trying to get you to think about aging, and this is me taking a component of that paper, redistributing what it's saying, and putting it on this slide for you. Okay? So let's go further and let's see what else this paper is going to try to evince to us. The pathobiochemical cause of aging is limited. Okay, so what do I mean of the cause is limited? So what does that mean? It means that our population currently experiences unprecedented long life expectancy, which implies that the incidence of age-related diseases like the neurodegenerative disease I just mentioned, AD, will increase dramatically as time goes on in any given population, right? So the pathobiochemical discussion of the cause is limited because you have limitation in lifespan. Okay. So therefore, there is a great need. All of us scientists want to say there's a need. That's how you get funded, and that's how you get a chance to research something. So there's a great need to more fully understand the aging process in order to circumvent those aspects, potentially, which initiate the neurodegenerative disease. So understanding aging, which is a separate process from neurodegeneration, you understand, at a systematic level is equally challenging, of course. That implies the best current research is to understand then what these people say, not organismal aging, but cellular aging. And we call that senescence. Now, why are they making this argument? And why am I guess, at least at first blush, agreeing with it? Haven't really thought about it, but I guess I agree with that. It's because we can get a handle on studying aging if we can study the cells. Very difficult to follow, individuals throughout their lifespan. For one thing, they're individuals, so we would need a lot of individuals to get a large enough N number to come up with any kind of induction at the end. Cells, not so limited. You can have millions and millions of cells packed into a milliliter of saline. So you can do a lot of things with cells. And so that makes sense, and I agree with it. Now, it stops there, though. When you study aging of cells, unless those cells are embedded in a living system, an organism, you're very limited to what you can say about that, even that process of aging, as it relates to sensu stricto, the entire organism. So you have to be able to look at both cells in test tube, cells in culture, and then also understand how those cells are functioning in tissues, how those tissues are functioning in organs, systems, how the organ systems are functioning in the entire living system, the body. And you can do that even in animals like rats and mice, which we can do a lot of fun genetic changes to via homologous recombination. We can do gene targeting, and we can knock out genes and look at what happens in the mice population, for example, over time. But then we're just studying mice and rats, or gophers, or foxes, or whatever other animal you want to be looking at. They may all be mammals, but they're not humans. So if you're looking at something like the central nervous system, and you're thinking about what we care most about that's affecting the central nervous system, such as neurodegeneration and therefore the losing of self, yeah, sure, we have models for that in mice and rats, but they're not anywhere near the complicated effort that you need to do to understand even a very small component of the human mind. So that's where pharmacotherapy comes in, right? Because humans are treated quite frequently with the pharmacognosy of what we have generated over decades now, hundreds of years, to be able to find chemicals that help us alter and change various processes, some of which seem to have an effect on the diseases that are most attributed to aging. Now, is that an effect on aging or an effect on the diseases, or both? Or neither? Square of opposition again. So for the brain, all of this I'm saying so far implies an understanding of how the molecular processes in neurons and glial cells change as we age. Right? And those are the two main types of cells in the central nervous system, neurons and glia. Also shows how there's an interaction of those cells affected by such changes. It's the interaction, the mechanisms of those interactions, which of course are perfectly fair game for biochemistry, pathobiochemistry, physiology, the kind of things we do here at Authentic Biochemistry. So I'm going to leave us here now because I want to develop the next stage of the lecture, which is going to involve a discussion of that paper that we just introduced, the Journal of Neurochemistry paper. I'm going to look at some data, And then we're going to look at probably 4 or 5 other papers recently published. And then maybe another 4 or 5 papers that support which we are the ones we are really doing a deep dive on, to be able to get our initial grasp on the diaventontology of the immune system and aging in humans, because I like to try to dedicate most of the things I discuss about the human population rather than, again, with model animals. Although, of course, there's going to be a time we're going to deal with model animals, and that's okay too. Right? So I'm going to stop here because I think it's a good stopping point. We've made the introduction, right? The prolegomena was much longer than getting into the first paper, but I think that was necessary because I don't expect you to go back and look at earlier lectures I gave, and especially like not synoptic ones like the one we just looked at on the autoimmune diseases and the neuroinflammatory responses in autoimmune diseases. But I wanted to give you that as an EG, as an example of where we're going to be going with aging. Okay. All right. So I'm going to stop here, and I'm going to say that this is Dr. Dan Guerra, coming to you from Authentic Biochemistry Studios on the wonderful first day of my most favorite month of the year, October. We're entering what I call October country. Hello, Ray Bradbury. So Dr. Dan Guerra saying bye.